All right. Welcome everybody to Research Expo Day 2. We are very excited to have all the audience members here and all the presenters from universities all over the country. We thank you so much for being here and supporting our fellow students who have put in so much hard work during this time of uncertainty into their innovative research projects. As a reminder, you will have the opportunity to vote for your favorite research presentation and award this audience's favorite at the end of day 3. At the end of today's presentation at 7 p.m., you'll be able to vote using our polling software for your favorite presentation. Additionally, our team will select 2 students from both days for 2 more awards called Best Presentation and Most Innovative Award. Best Presentation is awarded to the presenter who gave the most understandable, fluent, and concise presentation. And Most Innovative will go to the most innovative project that we had, such as it wasn't seen in society before and had a large impact on population health. Now, without further ado, we will go ahead and introduce our first presentation, Michelle Raja and Amal Umerani from the Telehealth Oncology Research Team. Thank you so much for joining us today, and take it away. You can share your screen whenever you're ready.
Okay. Should be sharing.
Thank you.
Right. Good evening. My name is Amal Umerani, and I am a sophomore at Emory University.
And I'm Michelle Raja, a junior at Stony Brook University.
Our presentation is on the effect of insurance type on cancer patient satisfaction with telehealth services during the COVID-19 pandemic. There is no primary data for our study yet, as we are awaiting IRB approval, but the purpose of our presentation today is to shine light onto the evolution of telehealth services during the pandemic and to describe the thought process behind our study. We have performed extensive secondary analysis to support the objective of our study. However, there is much more to learn within the field of telehealth related to health insurance and patient satisfaction prior to the pandemic, which is why we are exploring this area.
So, what are televisits? Televisits are the use of technology that allow patients to have real-time virtual visits with their healthcare provider. A prevalent but valid question that comes to mind regarding a televisit is the quality and satisfaction of telehealth care the same as in-person care? According to this 2019 study at Massachusetts General Hospital, 62% of patients found no difference in the quality of telehealth visits compared to in-person, and 21% of patients said that the quality of care was even better. Cancer patients have been a popular model population for telemedicine in the past due to many cancer patients being immunocompromised. Being immunocompromised puts these patients at a high risk to COVID-19. A study in Wuhan, China showed that 75% of patients who received chemotherapy developed a severe infection, while only 43% of patients without chemotherapy were infected. Telehealth visits have been advised to reduce the risk of exposure to COVID-19 to susceptible patients.
Now the question is telehealth beneficial beyond the pandemic? As we can see from the data from a study conducted in Australia, satisfaction with the telehealth model of care was proven to be high among patients and healthcare professionals. Benefits perceived included effective communication between patients and healthcare specialists, and reduced travel time and money expenditure. This data shows that telemedicine was effective before the pandemic. It is during the pandemic and can be effective beyond the pandemic.
Patient satisfaction is critical to good healthcare, regardless of its means of delivery, which is why it is crucial for us to study any possible disparities in telemedicine insurance policies which could result in unsatisfactory and unequal healthcare. Previous studies have found discrepancies on the rate of patient satisfaction depending on the type of insurance, that is, public versus private, used in a patient's in-person visit. Our goal is to investigate telehealth satisfaction in a similar manner. We would also like to compare patient satisfaction levels between in-person versus televisits and look into whether patients have a preference for one versus the other during the course of this pandemic.
Most, if not all, private insurances are offering some form of telehealth benefits in their current plans. Many states have encouraged health issuers to cover robust telehealth services. Issuers such as Cigna and Blue Cross Blue Shield have already taken steps to promote the use of telehealth services by providing expanded access to telehealth services. Further, public insurance under CMS has recently altered its policy so that virtual visits are billed in the same way as inpatient visits.
Additional changes have also been made to Medicaid since the onset of COVID-19. These include covering telemedicine even if the policyholder lives in non-rural areas and allowing physicians to connect remotely with their patients. While private insurance companies have also been expanding services and waiving telehealth copays, most issuers plan to stop these waivers by the end of this year. For instance, Anthem waived additional copays until September 30th of 2020, while Cigna waived theirs until at least October 31st of 2020.
Telehealth reaps many benefits, and the study hopes to provide a sustainable telehealth insurance model with high rates of patient satisfaction, and to expand to those who previously could not use them because of insurance coverage while reducing the exposure of immunocompromised cancer patients. This also makes the lives of those undergoing palliative care more comfortable by decreasing their reliance on caretakers. Transitioning to telehealth can lower the volume of patients at hospitals and clinics, which can be highly beneficial to immunocompromised cancer patients. Our primary data will be collected through an online anonymous questionnaire sent to various cancer clinics where oncology patients are treated. To eliminate recall bias, the survey should be conducted within 30 days of their last telehealth visit. In the survey, we will be using a satisfaction evaluation from the Telehealth Satisfaction Survey adapted for use in the Rural and Remote Memory Clinic to compare in-person and virtual visits. The survey questions consist of demographics, type of insurance, severity/stage of cancer, etc.
According to a 2019 report, the American Telemedicine Association identified 6 states and DC as states that have adopted updated telehealth policies and have passed parity laws. These states include Kentucky, Utah, Maine, Oregon, New Jersey, and Massachusetts. When a state passes a telemedicine parity law, it means private payers in that state have to reimburse for telemedicine care in the same way they would for in-person care. We pick states similar in policies, but we will study them as individual entities in order to eliminate bias. An analysis of a 2010 study conducted by the Rutledge Taylor and Francis Group showed that patients with Medicaid have a lower satisfaction score for patient-physician relationship. Specialists are also less willing to accept Medicaid patients than privately insured patients. Providers decline Medicaid insurance for a variety of reasons, which include administrative burden of navigating a state-run insurance program. Further analysis on telehealth can include possible trends within different states to deem if one state's insurance model is more effective than the others, or looking into specific telehealth services that patients wish for their insurance to cover, like phone applications, etc.
The rise of COVID-19 has forced us to examine whether a transition to telemedicine can be implemented if the right insurance policies are in place. The results of this study can lead to a sustainable telehealth insurance model that can give rise to highly satisfactory patient care. Telehealth is innovative, and it is the future of medicine. Thank you for listening to our presentation.
Thank you.
All right, thank you so much. You can now stop sharing if you would like. Thank you for your presentation. It was very informative, also very relevant in the context of the pandemic and healthcare, so we really like that. Now our next presenter is Sani Tripathi from the University of Arkansas.
Can y'all see me and hear me?
Yes.
Okay, so can I share my screen now? Or?
Yes, go ahead and share it now, please.
Okay, let me know if you can see it.
We can see it now.
Okay. Good afternoon, everybody. My name is Tripathi. I'm currently a junior at the University of Arkansas studying biomedical engineering, and it's an honor to be here. The research I'm going to be presenting today is entitled Identifying Cellular Senescence in Injured Kidney Using Imaging Mass Cytometry, and this research was done at Yale School of Medicine. So primarily the disease I focused on in this research was AKI, or also known as acute kidney injury. And it causes approximately 1.7 million deaths around the world, and it's the common cause of hospitalization amongst patients, accounting for between 8 and 16%. The disease can really affect anybody, but most cases that are seen by physicians are seen from patients that are 65 years or older. And the causes of the disease are kind of in three broad categories. The first is pre-renal, which is basically referring to the severe drop in blood pressure in the patient. Intrinsic renal, which basically refers to a direct injury to the kidneys from inflammation, blood clots, or other harmful toxins. And post-renal, which basically refers to ureter damage. And ureter is basically a tube which takes the urine from the kidneys to the bladder. And this can be caused from kidney stones or enlarged prostate in men. So the treatment of AKI is very limited at the moment, and it really depends on the specific cause and the specific case of the patient. But as a general use, most patients are put through hemodialysis, in which the kidneys are forced to basically pump through blood and remove harmful toxins. So when I entered the lab at Yale, a novel technology that has never been seen before, known as imaging mass cytometry, or IMC, was a technology that was being investigated that basically allows for the detection of more than 14 protein markers on a single tissue section. And the main purpose of the use of this technology was to better understand the composition of a human kidney, both in a normal state as well as a disease state, and give valuable information to researchers and physicians of the localization of certain protein molecules, of where they are being localized, the number and identity of various renal and immune cells that are present in the kidney. But also at the same time answer basic science questions such as which cells are dying in the process of acute kidney injury, how do these cells die, what injury pathways are they expressing, and which cells are being regenerated in the process. So the lab was creating a large antibody panel of more than 14 markers, consisting of various types of markers, including necroptosis, apoptosis, macrophages, and more. But specifically in my project, I really delved in the area of cellular senescence, and that is defined as cells that have undergone permanent and irreversible cell cycle growth arrest. And this can be caused by many different aspects, including inflammation, mitochondrial dysfunction, and oncogenic mutations. And this can be very detrimental to the patient when accumulated in the kidney. So basically the cells essentially stopped dividing in the stage of mitosis. And within cellular senescence, there are different inhibitors, but the main inhibitor I focused on in this study was P21. And through literature, it is known that the upregulation of P21 can cause cellular senescence and blockage between the G1 and S phase of mitosis. So in this study, I basically used an in vitro system to really find out more about the upregulation of P21 and how it is reacting to the kidney cells itself. So in my study, I gathered HK2 cells and cultured them in 4 and 8 chambered slides and allowed them to reach full confluency after 3 days. And the reason why I did 3 days is to make sure that the cells were there was not enough medium to where it is too spaced out between the cells, but not too much cluttering of the cells to where they're stacking on top of each other. The cells were treated then using cyclosporin A, which is a known drug to induce cellular senescence in kidney cells. And those are treated, I treated those cells for 24 hours at various concentrations, including 5, 10, and 50 micromolar. After the 24-hour treatment, immunostaining was performed of these cells using the P21 antibody at various dilutions, including 1 to 100, 1 to 300, and 1 to 500. And the results were observed using immunofluorescence imaging. So as you can see here, you see the first two set pairs of images. So you have when you do immunofluorescence imaging, you have three channels. You have blue channel, which is a DAPI channel, which indicates the nuclear staining. You have the red channel, and then you have the green channel. For my study, I only use the red channel because the red and green channel are used for two different markers. But for my case, I was only using P21, so I only did red channel. So these first two sets of images indicate, firstly, the nuclear stain that the cells were present and not dead when I took this image. And then the last two images, as you can see, is completely black. And the reason why that is because this image only indicates, the first black image only indicates staining of the primary antibody, and then the second image only indicates the secondary antibody. And the reason why I did this in the study and included it in the poster is to make sure and to validate that the antibody that we were testing was specific to the cell that we were targeting. And so due to this fact, it did in fact, as expected, turn all black. And then the last two images are comparing the control group and then the treated group after cyclosporin A had been applied. And so as you can see, there's a significant increase in the signal that was obtained, a stronger red signal. And specifically, we found that the P21 antibody was specifically localized in the cytoplasmic region of the HK2 cells, which is an incredibly important finding. And this confirmed that, firstly, that our binding was specific, and secondly, that each cell was targeted at a cytoplasmic level in the cell. And then also after this, we wanted to further confirm our findings. And so we performed qPCR as well to evaluate the expression levels of P21 that was being tested. I did not include this in the poster, but in fact, after qPCR, the expression levels were increased, which validates our study findings from immunofluorescence as well as the literature findings as well. And so looking forward, the future work that could be expanded on this is to potentially include this in the antibody panel that was being tested from the lab itself. So to do this, we could use this antibody concentration as well as treatment strategy in the IMC panel, in the sense that we can apply it to human kidney tissue that was being obtained from biopsies from patients. And to do this, specifically for IMC, the antibody would have to be tested firstly on the tissue and then conjugated to a heavy metal isotope and then evaluated through an imaging software tool. So as an example, you can see on the right, the left-hand side, that you can see a kidney biopsy specimen and then the various different colors of markers that are being illuminated in the kidney anatomy itself. So based on my findings, this could be also applied in the sense. And this is a very novel piece of technology that has never been done before in kidney and can be translated directly into the clinic in terms of testing a patient for multiple causes of AKI instead of one at a time and going through the laborious process of going through the pathology report and then going back and forth. And really, this could expedite the extraneous process itself. But thank you guys for your time and attention, and it's an honor to be here again.
Thank you so much for your presentation. That your project is very innovative, and we are very thankful to hear from you today. Now we are going to transition to our next presentation. Yancey DeWayne Williams II from the University of Alabama at Birmingham. Feel free to share your video and your screen and take away your presentation.
All right, thank you for having me here.
Of course.
All right, I'm just sharing my screen right now.
Your audio is a little low. Could you please speak up during your presentation?
How's my audio now?
Oh, that's perfect. Thank you.
All right. Can everyone see my poster?
Yes, we can.
All right, I'll begin.
If you're able to zoom in certain parts during your poster, that'd be great. If not, no worries.
Oh, I'm sorry, I can't, but.
That's okay.
Okay. Hello, everyone. My name is Yancey DeWayne Williams II, and I am an undergraduate sophomore majoring in biomedical engineering in the Science and Technologies Honors Program at the University of Alabama at Birmingham, or UAB. I have been working with my mentor, Dr. Alexa Mathesis, and my graduate mentor, Tom Narwell, on this project, modeling simultaneous two-wavelength axial radiometry, or also known as STAR, microscopy, to study endocytosis. So first, let's try to understand what all that means. Endocytosis is the internalized process of material such as nutrients and other molecules that are too large to pass through the membrane into a cell, the surface of the membrane, using coated vesicles. The main ones that we're going to be looking at today is clathrin. And these clathrin vesicles can be used to form different components of the membrane. And the pathway is very highly dynamic and versatile as to going to different parts and to cargoing on the different parts of proteins and other molecules that pass through the membrane. Nevertheless, clathrin independent entry portraits have been described as vital process, especially in mammal cells, as an intricate part of life. So one of the simplest ways in order to see the endocytosis is a process called axial resolution, which is a type of microscopy that takes advantage of the excited fields of the different molecules in the cells, and they are able to come back using different wavelengths and intensities of the light. So the aim of this project is to design a MATLAB code that will model theoretical measurements of the vessel's growth during endocytosis acquired through the STAR microscopy. Furthermore, STAR is used as a comparison between theoretical and experimental data that was performed in the lab here at UAB. It is very unnecessary to precisely understand the biophysical properties of membranes, dynamic function, and the formation during clathrin metastatic endocytosis. So the first part of the project was to create the MATLAB code, which gives us the Z position, which is the position of where it is in relation to the entire cell of the protein. And with the given equations of earlier research, we were able to find the initial and final intensities and wavelengths of the fluorescence that was used to shine upon the cell to find out each certain molecule. Next, we simulated these endocytosis by recording the data of the microscopy using real-time fluorescence imaging with about 20 nanometers axial. And then we would modify the MATLAB program in order so it to reflect current and previous data of endocytosis and to record a 3D model of the protein during this entire process. And finally, in the future, we plan on modifying this MATLAB code to predict a 3D model as well as to create a different type of microscope in order to accurately get these findings. So if you look into the computational data, this is just a snippet of code that I've used in the lab to record the intensities and wavelengths of the different molecules. And you'll see positions on D1 and D2 just to see what the relation is between two different molecules within a cell. And we use that to using the theta value of how the light is shined upon the cell, and the intensity values is calculated through the equation shown in the presentation. And then you would see the DZ, which is the change in the Z position, just to see how far it moves and how does it change throughout this entire process. And the last bit shows the plotting of the graph of delta Z and the total intensities, just to see if there's a relationship, which there should be. In figure 2, you will see that there is a clear correlation between the theoretical and experimental. The red is the experimental and the green is the theoretical. So you do see that while they are close enough, that we would like to try and make our MATLAB code better in order to improve upon this. So where do we go from here? This START technique is very revolutionary because it takes a very basic concept of just looking at cells through a microscope and actually changing of how can we make this better. So what we're planning on doing with this research, instead of continuing to use a microscope by changing the theta value of the degree of how the light is shined, of using a 360 microscope in order to show the cell in its 3D form instead of just flat on a plate with the microscope in order to show just the face value. So for the implication of this research, the real-time fluorescence imaging can be used to detect the different angles of the proteins and the curvature in order to see the 3D model of the cell during endocytosis. This method employs the protein as it is tagged on by two different fluorescents and image to obtain the different ratio between these two channels. And we believe that this approach will be revolutionary to visualizing the nanoscale organization of molecules and endocytosis of the growth factor. I would like to take this time to thank Dr. Mathesis and Tom Narwell for helping me with this project, as well as the University of Alabama at Birmingham for giving me the Presidential Summer Fellowship Grant to complete this research, as well as the Department of Biomedical Engineering and the Department of Cellular on Developmental and Integral Biology. Thank you, everyone.
All right, great presentation, Yancey. You had some outstanding presentation skills and explanations, so thank you so much for that. Our next speaker is Monge from Rensselaer Polytechnic Institute. Hey, Molly.
Hi, I'll share my screen.
Perfect.
Okay, thank you for the kind introduction, Alejandra, and thank you all for coming here today. I'm really excited to tell you about my research. My name is Monge, and I'm a rising junior at Rensselaer Polytechnic Institute, where I major in biochemistry and biophysics. The research I'll be talking about today was completed at the Wadsworth Center, New York State Department of Health, and I'm really excited to get to tell you about bacterial genetics and regulating gene expression. Bacteria need to be able to control the transcription of certain genes to respond to environmental stressors. When we think about how scientists have mainly studied gene expression regulation in the past, they've really mainly focused on the initiation of transcription. We know that RNA polymerases can use different sigma factors and different transcription factors to transcribe different genes. The onset of genome-wide analysis has allowed us to really take a deeper look inside bacterial genomes. And in particular, we've been able to look at the termination of transcription. And my project studied the termination of transcription in E. coli. There are two different mechanisms that E. coli can use to terminate transcription. The first mechanism is known as intrinsic termination. In intrinsic termination, RNA polymerase will transcribe a GC-rich region followed by a poly-U track. That GC-rich region is very affinitive for each other, and they'll fold up and form secondary structure. Secondary structure is simply RNA forming structures that are not linear. And this, let me get my pointer. Okay, this loop right here is known as a hairpin loop. And RNA polymerase's interactions with the DNA will be weakened, and that will cause RNA polymerase to come off and transcription will be terminated. There we go. And today we're going to be talking about the other type of transcription termination known as Rho dependent. Rho is a hexameric ATP-powered helicase that will bind to RNA in an open conformation at something known as a RUT site. A RUT site stands for Rho utilization site. When Rho binds, it will begin translocasing along the RNA strand until it eventually catches up to the RNA polymerases. Conformational change happens, and the transcript is released, and termination is completed. When we think about transcription termination of any type, we would think the termination must be occurring someplace at the end of the gene because we want a properly folded protein and we want a fully functioning gene. So normally during transcription, translation is happening concurrently in bacterial systems. So Rho is not present because the ribosomes are, and they're blocking Rett sites, so it can no longer bind. In the case of the genes that I studied, Rho was terminating prematurely some places in the 5' untranslated region and the gene. And my goal of my project was to validate genome-wide analysis that pointed towards this phenomenon happening. And this was really exciting work for my lab because it had never been documented at this wide of a scale. And now I'm going to get into some of the experimental design on how I found the genes that were terminated prematurely. I created 2 different fusions for this project, both the short fusion and the long fusion. The reason why we had 2 different fusions was to figure out where the approximate location of transcription termination was happening. If it was going to occur in the 5' untranslated region, you would see a change for both the short and the long fusion. I'll talk more about that change on the next few slides. And if it was going to be occurring in the long fusion, you would only see that, if it was going to be occurring in the open reading frame, I have that denoted the ORF, it would be occurring only in the long fusion. And I just want to point out that we used a reporter gene in this experiment known as LAC-Z. And LAC-Z, as a reporter gene, it gives off a phenotypic effect that we can measure, and it produces a yellow color that we can quantify the absorbance. So in the wild type strain, Rho is completely functional, so it should terminate someplace in the 5' untranslated region and the gene. And when that termination happens, you would expect very, very little yellow color because LAC-Z cannot be transcribed or translated. In the Rho mutant strain, its quality is much more diminished. So the termination is going to occur after the gene, and you'd expect a lot more yellow color. So this is how we were able to determine the approximate location. There were 2 non-terminated genes within my study. As you can see for both of these genes, the activity didn't change for either the wild type or the Rho mutant. So while we expected for the wild type something like this to happen with only a little yellow, and something like this to happen with the Rho mutant, both of these had a lot of yellow both times. So that's how we know these were not Rho terminated. First classification of genes that we'll talk about are those terminated in the 5' untranslated region. For this to happen, again, determination must be occurring in both the short and the long fusion. First, we'll look at SPA. So I labeled both of the 2 segments S and L for short and long, just for easier clarification. So you can see for both of them in the wild type, there's very little activity, and that activity corresponds to that yellow color. You can see that when Rho is mutated, that activity skyrockets. This trend was seen for all the other genes here. And the reason why we, again, classified these as being terminated in the 5' untranslated region was because the change occurred in both the short and the long fusion. For those occurring in the open reading frame, which for those of you who don't know, open reading frame is just another way to say the coding region of a gene, we would only see that change in the long fusion. So looking at MOA-A, you can see that in the short, there's no statistically significant change between the wild type and the Rho mutant. There's that sharp increase in the long. And this was similarly shown for each of the genes here. And SUG-E is probably one of the best examples because there's very little to no change when the 5' untranslated region is only included. But when you include the open reading frame, that steep increase is shown. And again, because this occurred in only the long fusion, we know that this is in the coding region. So this was really exciting. And the lab has been trying to figure out like what's going on now and why is this happening. One possible explanation is that some type of protein is binding upstream of a Rett site, and when it binds, the RNA can undergo a conformational change and a Rett site can become exposed. Again, because RNA can form that secondary structure, perhaps there's a Rett site being closed when nothing is bound, but when that protein binds, it triggers a downstream action. And when we look at this other one, I know I mentioned in the beginning that ribosomes could be blocking Rett sites, but perhaps something called a small non-coding RNA could be binding to that ribosome binding site, and it would open up another Rett site similar to the protein, but this time in an RNA form. And with that, I'd just like to thank my lab. I really, this was a great experience for me. The environment was very collaborative and I was treated like a colleague, and everybody was so supportive of me. I'd like to thank Joe Wade, he was my mentor, and particularly my lab mates, John Plicknick, Carol Smith, and Gabby Van Ulite. I'd also like to thank Matt Cohn. He was the director of our REU program, and he really organized everything for us and made this experience possible. And lastly, I'd like to thank the National Science Foundation for funding this research experience. Thank you for your time.
Thank you so much, Molly, for your presentation. It was very understandable. I loved how you used mostly images throughout your presentation, and it was a series of slide-by-slide explanation. I thought it was wonderful. I took genetics while I was an undergrad, and they actually refused to teach this part of termination because they said there's very little research about it. So I'm very grateful to hear from you and the research you've done about this. So thank you so much.
Thank you for letting me present.
All right. And our next presentation today is Jennifer Smetanick from Arizona State University. Thank you so much for joining us, Jennifer. Feel free to share your video and your presentation whenever you're ready.
Yeah, I'll get that up right now. One second. All right, are you guys able to see it?
Yes, we.
Awesome. I'm really excited about this project, and I'm really grateful that I have this opportunity to share with you guys. If you have any questions along the way, definitely put them in the Q&A. All right, so my project focuses on neuromuscular diseases and a potential therapeutic application using a chemogenetic tool to aid in neuromuscular junction recovery. Now, I know I said a mouthful right there. Let me go and break that down. So neuromuscular junctions, or NMJs, are the synapse between the lower motor neuron and the muscle fiber that regulate muscle contraction. Now, what we see in muscular dystrophy is the progressive weakness and loss of muscle. Due to the degeneration and regeneration of muscle fibers during the course of muscular dystrophy, NMJs are significantly disrupted. We're interested in examining the morphological changes in the NMJ of a mouse model with muscular dystrophy. Moreover, we're attempting to develop a tool to test whether stimulating lower motor neuron activity in vivo has any therapeutic application within muscular dystrophy or any other neuromuscular disease like ALS or SMA. So to manipulate lower motor neuron activity, we generated a G sub Q DREAD mouse model to study this. Now, DREAD stands for Designer Receptor Exclusively Activated by a Designer Drug, and that is a chemogenetic tool that can manipulate signal transduction pathways. So when G sub Q DREAD is activated by its said designer drug, CNO, which stands for clozapine oxide, increased neuron firing should occur. One aim of this study is to provide a proof of concept that verifies expression of the G sub Q construct within the lower motor neurons. Second, we examined whether CNO administration leads to activation of motor circuits. Just to kind of summarize that up pretty quickly, chemogenetic tools are a type of receptor that engineers create to be opened or closed by a new type of drug that's created. So this is not a naturally occurring type of receptor. And so because we can now manipulate how it is opened or closed, we're trying to determine if we open it, can we have more lower motor neuron activity that can potentially rescue muscle fiber, muscle movement, muscle contraction. I think that's such an important question to ask because so many people are affected by neuromuscular diseases, especially children. So that was something I was really interested in studying, and I'm so glad I had the opportunity to do so. Okay, so now on to methods. So to better visualize the concepts I've just discussed, we use immunohistochemistry to analyze the histology of diaphragms of muscular dystrophy mice and spinal cords of G sub Q DREAD expressing mice. These samples were then imaged on a confocal microscope and then analyzed further with Adobe Photoshop and ImageJ software. So to answer the first question concerning NMJ morphology within muscular dystrophy mice, we immunostained whole mount diaphragms of wild type, control mice, and then the muscular dystrophy mice. So in the green, here and here, are the neuromuscular junctions, and they're labeled with bungarotoxin. A typical neuromuscular junction is described as having a pretzel-like shape. So this is kind of a pretty good representative of what NMJs look like. One let's see. So yeah, and then below here are the individual representations of each genotype. So I have 2 pretty good representations of the wild type and then 2 of the double mutant. These are 2, like they have the same pixel size, the number of pixels in each image. So the bigger size we see here is representative. So just from looking at these images, we can conclude that the NMJ morphology is in fact altered in muscular dystrophy mice. Mutants exhibited a fragmented and dispersed phenotype. As you can see, there's more number of particles. So further quantification of NMJ area and number of particles using ImageJ software shows a statistically significant increase in mutants compared to wild type. So the red is the mutant, black is wild type. And to be as unbiased as possible, in our ImageJ software, we use the exact same settings, brightness, number of pixels, everything to make sure it's counting the number of particles in as an unbiased way as possible. So now that we've kind of confirmed that the NMJ morphology is in fact altered, we can move on to our second mouse model that uses the chemogenetic tool. I'm gonna zoom in because the next images are a bit smaller. All right, so on to this result. So the first step is to verify co-localization of the G sub Q construct within lower motor neurons. That basically is saying that we want to make sure that construct that we are putting into the subject is going to the right location. And that right location for neuromuscular diseases is lower motor neurons. So to accomplish this, we immunostained lumbar section spinal cords. In the green here is where the G sub Q construct is. It's labeled with GFP, so these brightly lit green. And in the red is where the lower motor neuron activity is, or lower motor neurons are. That's labeled with chat. So ImageA just shows an overlay of all 3. So this is all the same image. I just separated the different channels out so you can clearly see where the green and then the red is individually. The blue is just DAPI, that's DNA. So not important for this specific thing, but A is the overlay of all of them. So ImageA shows that the G sub Q construct and lower motor neurons are colocalized within the ventral horn, which is exactly what we needed before going to the next step. So that just confirms the DREAD receptors being expressed in the correct location. So now that we have that, we can go on to our second section using the same DREAD mouse model, which was to examine the effects of CNO administration in the G sub Q DREAD mice. Like I said before, CNO is the designer drug that is connected with this specific DREAD model. So on the left, we have a lumbar section spinal cord of a mouse expressing the G sub Q DREAD receptor. And then on the right, we have a section of a mouse expressing the construct with the CNO administration. Like before, the green is labeling for the G sub Q receptor. So you see here and here. And then red is now labeling for FOSB, which is an activity-dependent gene. So the mouse with the CNO administration has noticeably more FOSB expression. Now this is a bit smaller. I'm not sure if you can see on Zoom, but there's some sparse dotting of FOSB here without activation. And with activation, you can see it's in a much more broader area, spotting around there. So this confirms that CNO is binding to the G sub Q DREAD receptor and increasing neuron firing. That's really important. That's showing that this engineered receptor we created is acting accordingly to how we wanted to increase the neuron firing, especially in lower motor neurons. Zoom out really quick. So in conclusion, NMJ morphology is significantly disrupted in muscular dystrophy mice, and the G sub Q DREAD tool is functional because of its co-localization within lower motor neurons and increased neuron firing when activated with its designer drug, CNO. This chemogenetic tool may have potential therapeutic applications in rescuing function within neuromuscular diseases. Further directions include testing if lower motor neuron activity firing can be rescued with, what was I saying? Future directions include testing if increasing lower motor neuron firing can rescue those morphological changes seen in muscular dystrophy mice, NMJs. I want to thank my lab, Banner Research for helping with this project, as well as the National Pre-Health Conference for allowing me to speak here. If you guys have any questions, please feel free to leave them in the Q&A. I'm really excited about this research, and I think it has so much potential to help, a lot of children who have muscular dystrophy, problems, other neuromuscular diseases like cerebral palsy and that kind of stuff. So thank you again. That concludes it.
All right. Thank you so much, Jennifer. Great presentation skills. I can feel that confidence, and also you simplified everything for the audience, so it was pretty accessible to everyone. Thank you so much.
Thank you.
Now, our next speaker is Jimena Collado from Northwestern University.
Hello, everyone. Hello, Alejandra.
Hello.
Let me start sharing my screen. All right, can everyone see it?
Yes, we can see it.
All right, fantastic. So thank you everyone for joining. Today, I will be presenting my research on abnormal nuclear morphology, its relationship with DNA damage and nuclear protrusions. So let me start by stating why abnormal nuclear morphology is relevant for us and why it is very crucial for us to understand it, especially within the field of health and medicine. So abnormal nuclear morphology is characteristic of many diseases, including heart disease, diabetes, and several different types of cancer, to name a few. It is a very well known and very recorded phenomenon, yet it's not very well understood. It is in fact used as a pathological marker in diagnostic tests, such as the pap smear, which is this image that you're seeing right now on your screen. And the pap smear is used to detect cervical cancer. It's used as a diagnostic test. And in this test, they observe whether or not your cells present a nuclei with abnormal morphology. So this is just one of the examples of how abnormal morphology is very prevalent in health and medicine. And two key components that I focus on regarding abnormal morphology in a diseased cell is the chromatin organization, the chromatin compaction, and blebs, which are a form of abnormal nuclear morphology. So let me give you a little bit of background on how chromatin is compacted so that you're able to better understand this research. So chromatin can be compacted in two ways. Remember that the DNA is wrapped around a histone core 1.65 times, and there is going to be histone tails popping out, as you can see in this images right here. And these histone tails can either be methylated or acetylated. If they're methylated, then they're going to be, you're going to get this chromatin compaction. So it's going to be a low gene density, very high compacted form of chromatin, and this is going to be termed heterochromatin, which we have right here. On the other hand, we can have the decompacted form of chromatin, which is acetylation on certain histone tail areas, and this is going to be euchromatin. And this is going to be very gene rich because the DNA is accessible. So now you may be wondering, well, how is chromatin organization and chromatin compaction related to morphology? Well, it was recently proven by my mentor, Professor Stevens, that you can decompact chromatin and induce blebbing. So you can induce blebbing solely by decompacting the chromatin. And as you can see here, this is a bleb nuclei, and bleb are these nuclear protrusions coming out of the nuclei as shown here. And a way you can induce blebbing is through treatment with valproic acid. And valproic acid is a drug that decompacts the chromatin and increases euchromatin, as thus it's going to induce the blebbing. And we can see that in this graph right here. Another thing that we observed in these bleb cells is that they showed significant amounts of DNA damage. And this is as we measured by gamma H2AX, which is a marker for DNA damage and repair. And we also observed that the nuclei, the bleb nuclei, were very likely to rupture. They were 80% more likely to rupture than normal cells. So we are observing these three phenomenon in this nuclei, right? We're observing DNA damage, we're observing blebbing, we're observing rupture, but yet we don't really know how these are related. It could be that DNA damage is inducing the blebbing. It could be that the blebbing is then inducing the rupture, but we don't really know for sure how these three things are related. So that's what my research is about. So it's going to test this, is the DNA damage inducing the blebbing? And my hypothesis was that an increase in DNA damage will cause an increase in nuclear blebbing. And this is because the DNA repair machinery needs to access the genome in order to repair it. So you may be thinking that the chromatin needs to be decompacted for the repair machinery to do so. And as we previously saw, the repair machinery, if the nucleus is decompacted, then that's going to induce blebbing. So in order to test this hypothesis, we induced DNA damage with DNA replication inhibitors, hydroxyurea and ampicodylin, which are two drugs, on mouse embryonic fibroblast cells. And then we used immunofluorescence to visualize DNA damage and nuclear blobbing. We again used the gamma H2AX foci. The gamma H2AX again is the protein, DNA repair protein that tells us where the DNA damage is. And we used a Hoch stain on the nuclei to be able to visualize the blobbing as you see here. So how did we quantify these sites of DNA damage? So we used an imaging software, ImageJ, and all of these bright dots that you see here, all of those bright dots are a site where the gamma H2AX protein is actively repairing a DNA damage site. So that's how we know there's DNA damage there. And we can pass an intensity plot for one of these foci, and we're gonna get a graph that looks similar to the one right here in the middle, which follows a Laplacian distribution and a Gaussian function. So then we can calibrate the software to automatically select all of these points on the image that follow this same distribution, this same function, and the software can automatically count all of the DNA damage foci in the nucleus. So what do we find after measuring all of these nuclei, its blobbing and its DNA damage? We found that DNA damaging drugs do not induce blobbing. So here you can see that the two drugs that induced the DNA damage did not cause a significant amount of blobbing. So if our hypotheses were true and DNA damage did induce blobbing, then we would expect APH, which is the one that caused the most significant DNA damage, to also cause the most significant amount of blobbing. But it did not even produce a statistical significant difference in blobbing. So what this tells us is that the most likely pathway and relationship between these three components looks like you get the chromatin decompaction, which induces the blobbing, which is going to induce the rupture, which is then going to induce the DNA damage. And we have correlational data corroborating that DNA damage is inducing the rupture, that the ruptures are inducing the DNA damage, but we need to prove this causation. So we can do so by live cell imaging with nuclear ruptures with NLS-GFP, which stands for nuclear localized GFP. GFP is a green fluorescent protein, and we can also use the DNA damage marker mCherry53VP1, which can be endogenously expressed in the cell. 53VP1 is another protein of the DNA repair machinery, and mCherry is just a red fluorescent tag. So we can be able to see here the NLS-GFP is what's shown in these videos. So you're able to tell when the signal is being spilled into the cytosol. So when is the nuclear rupture happening? Right here, you can see it. And then you're able to measure these sites of intensity, the 53VP1, and determine whether the DNA damage increased before or after the rupture. We most likely expect it to be after the rupture. And in fact, it makes a very much sense that the rupture is the one inducing the DNA damage, because once you rupture, you basically lose the entire purpose of a nucleus, and there's DNA damage repair factors spilling into the cytosol, and your ability to repair DNA damage is of course impacted. You also have proteins coming into the nucleus, interacting with the DNA in ways that they were not meant to interact, so that can also cause DNA damage. So this is most likely what we think the pathway looks like, and our future directions are proving that it is the rupture that's preceding the DNA damage. So thank you everyone, and I would like to thank especially my research mentor, Professor Stevens, who is currently at UMass, my PI for this project, Professor Marco at Northwestern, my academic mentor, Luke Flores, and and Dr. Bennett Phillips for providing the funding to make this research possible through the program Physical Sciences in Oncology. Thank you.
Thank you so much, Jimena, for your presentation. It was very, very intriguing and interesting. And it was very interesting how you could visualize that chromatin decompaction and compaction. So thank you so much for sharing.
Thank you. And I'll answer any questions on the chat right now.
Perfect. Up next is our last presentation before we vote. We have Amesh Sarecha from Columbia University. Thank you so much for joining us today. Feel free to share your screen whenever you're ready.
All right, could you guys see my screen?
Yes.
All right. Thank you so much for the kind introduction. My name is Amesh Sarecha. I'm a junior at Columbia University studying biochemistry and mathematics and my project today is the role of the hepatic stellate cell on metabolic diseases. So you may ask, what are hepatic stellate cells? So in the liver, there are 4 types of major cells. We have hepatocytes, which are the chief functional cells of the liver, and they comprise of about 80% of the cells. We have Kupffer cells, which are specialized macrophages, which perform the phagocytic duties. So they help break up toxins and molecules that the cells doesn't need. We then have the endothelial cells, which they form the blood vessels and carry blood throughout the liver. And finally, we have the cells that are of interest to us today, which are the hepatic stellate cells. And these are mesenchymal cells that play vital roles in liver physiology and fibrogenesis. And they contribute about 5% of the cells in the liver. So why we chose to study the hepatic stellate cells is because hepatocytes, Kupffer cells, and endothelial cells from past literature we saw have largely been studied in the progression of diabetes, a major metabolic disease. So over here on the right is just a stain of all the 4 cells I just mentioned. And what is diabetes? So we all may know a loved one, friend, family that has diabetes. It's basically a metabolic disease characterized by high blood sugar levels over a prolonged period of time. And this is otherwise known as hyperglycemia. Why hyperglycemia is such a serious condition is because it can cause defects in our vision, our heart, and all of our major organs. And diabetes consists of a few categories, but it can basically be broken down into 2 categories. First one being type 1 diabetes, which results from the pancreas failure to produce enough insulin. And this is because the beta cells, which are the insulin producing cells in our liver, they're basically wiped out. And this is an early onset of a disease, which usually occurs in childhood. Then you have type 2 diabetes, which is a later progression, and it results from insulin resistance. What this means is that the body fails to respond to the insulin, and that leads to the sugar being able to stay in our bloodstream and not being absorbed by the cells, leading to hypoglycemia. So to give some statistics, this diabetes has a really big impact on population health. Currently, last year in 2019, it's estimated that there were 463 million people that had diabetes, and that costed us $825 billion. And there's epidemiological studies that show that this number is projected to increase by 2030 to a total of 578 million, and then in 15 years later to a total of 700 million people. Needless to say that this is a chronic metabolic disease that everyone would benefit for if there was a better cure or better treatment. Because to be quite honest, with the current treatment now, and even if there is treatment, diabetes leads to sometimes death. So what we did, we wanted to study more about the disorder, and we wanted to take a look in particular at how hepatic stellate cells would have a role in diabetes. So from prior research, we know that they're non-parenchymal cells of the liver, and they have an established role in fibrosis and tissue repair. We also know that they express multiple stem cell markers, and they can differentiate into various cell types. In the past few decades, stem cells have been a large interest for people because they can basically regenerate tissue, and they can be determined by their potency, which they can be totipotent, pluripotent, or multipotent, which means that they have the capability of that basically accounts for how capable they are to differentiate into cells. So from literature, we saw that the hepatic stellate cells can successfully differentiate into insulin secreting cells in vitro. So we found that to be very interesting because if beta cells are the cells that produce the insulin, and if we can have our hepatic stellate cells produce the beta cells now, our body could get rid of this condition known as hyperglycemia. But based on this, we wanted to tackle the problem on a larger scale now. We wanted to answer the question, how is paracranial communication altered when you remove the stellate cells? So in order to do this, we basically had a knockout mice model where we removed the stellate cells and we compared that with normal mice that were fed under a controlled diet. They had normal pancreas pathology, and all the mice were maintained under normal laboratory conditions. They were fed at the proper time. We were able to monitor their body weight, the water intake, and they had access to clean drinking water throughout the study. So the the mouse tissue samples were formalin fixed and they were put in paraffin embedded blocks. And we were able to take those whole cell extracts that we had and subject to a simple BCA protein assay, which was able to determine the protein concentration of our protein of interest at the time. And we then were able to resolve the protein extract using SDS page for immunoblot analysis, otherwise known as Western blotting. And we were able to estimate the protein levels, and we compared the protein levels that we found to a housekeeping known as beta-actin. What this means is we use beta-actin as a control to compare our enzymes and proteins to see how the values changed. And our most significant find after many trials was that liver de novo lipogenesis decreased in the hepatic stellate cell depleted mice. And what this means is that the liver synthesizes new fat from mainly carbohydrate resources. Before we were like proud of this and happy we found a result like this, we wanted to make sure we actually did a proper knockout of the hepatic stellate cell. All the way to the right, it shows that we have over here a marker called desmin, which is present in the hepatic stellate cells that was completely knocked out, because from here you could see it was present here and not present here, which led us to see that our knockout was good. ATPCL in the middle is a key enzyme of liver de novo lipogenesis, and we found significant levels of reduction in the mice. We also saw that a protein over here, ChRABP, which stands for carbohydrate response element binding protein, decreased as well. And that's a glucose-mediated transcription factor that strongly regulates glycolytic and lipogenic pathways. So we had to wipe out 2 of our samples because they were not working properly and there was some problem, but we also wanted to test for more key enzymes. So we tested for a bunch of these enzymes all the way here at the right, and we saw another significant find that ERK decreased in the hepatic stellate cell mice. Again, since we were using the same samples, we had to wipe out these 2 columns. And what the ERK signaling pathway was, it seemed to play a role in kidney fibrosis and accelerated by blood glucose fluctuations. So we saw that this could be a very interesting find. We had additional findings such as the enzyme AKT473-FOS decrease, we had PPAR alpha decrease, and PPEC increase. While before I told you what the findings were for those key enzymes, these key enzymes are part of many pathways downstream of different things, so we were unable to correlate what this would do. However, one thing that we did find that helped to corroborate our finding of liver de novo lipogenesis is that ATGL increased, which is very good because since ATGL increased, it basically means that lipolysis increased, which is the opposite of liver de novo lipogenesis. Over here is just a graph of the quantification of the Western blot we had, and it's normalized to beta-actin with all the genes. And in conclusion, what this meant for us was that increased rate of liver de novo lipogenesis, it appears to be combined with a multitude of metabolic diseases. And the most prominent of that is non-alcoholic fatty liver disease. And that's basically where fat builds up in the liver, which shows evidence of hepatic steatosis. And why this is a very important disease, and a lot of people all over the world have undergone this disease, it progresses into something known as NASH. So that's fibrotic hepatic inflammation, and that can worsen into cirrhosis, which leads to carcinoma and cancer. And our results help to show that reducing de novo lipogenesis through the stellate cell deletion may reduce the chances of contracting non-alcoholic fatty liver diseases, because typically de novo lipogenesis believes to contribute to the pathogenesis of this. So our future directions help to point at the better understanding of if it could really be a good candidate for reducing non-alcoholic fatty liver disease. So we decided to perform qPCR in the future to account for the DNA differences, which would support our Western blot data. And we plan to do immunohistochemistry, or iDisco, which is basically where we check for the localization and presence of fatty liver on a 3-dimensional level. And finally, we are interested in doing a glucose tolerance test where we would monitor the glucose of the mice over a certain period of time. And this would point to maybe other metabolic diseases that we would be interested in too. And I would like to give a special thank you to my PI, Dr. Christopher Buettner, and my amazing mentor, Dr. Kenichi Sakamoto, and the Icahn School of Medicine in Mount Sinai. Thank you so much.
All right. Thank you so much, Amesh. Great presentation. And now it is time to go into the polling. So I will turn that feature on and make sure that all of you guys vote for your favorite presentation of today. And this is based on what the audience decides. And we will let you know who won